So in short, the gene expression pattern, for example, can be associated or can be integrated with the clinical data, and this can help us identify molecular biomarkers for diagnostics and for therapeutic selection. And new discoveries include better understanding of complex diseases at the subcellular level, as well as it can help us in the target identification for novel drug design. It can also help us repurpose existing drugs for different purposes. One of the very famous example is the usage of metformin drug, which was traditionally, or which is still used to treat diabetes, is actually getting repurposed to treat clinical cancer data, or repurposed to be anti-cancer drug in certain conditions based on the omics data analysis. So what this actually creates is, this creates a disconnect between, for example, the data that is obtained from the lab technicians and clinicians, and the data that can be processed and understood from the biomedical research ecosystem based on the data analysis method and data analytics that we can perform using bioinformatics. This disconnect actually breaks up the cycle of innovation due to lack of data-driven research skills among the otherwise very effective biomedical personnel that we have in dealing with increasing demands and increasing changing in the unpredictable landscape of the omics data that is currently present. So to fill this gap, what we need to do is we need to develop a more for example, ecosystems that are prone to be decentralized and that are prone to be data-driven approach, right? And we have been given a taste for this current scenario from thanks to the pandemic, or not thanks to the pandemic. This has already started to affect healthcare, and more subtle trends of disease management and drug development are also being observed, right? And these industries are most affected as going decentralized for laboratory has been found very difficult, and several harsh COVID protocols has to be taken to enable working inside this laboratory environment. But on the contrary, data-driven approaches in large-scale projects can be performed in a decentralized manner, and this is key to the future of clinical and biotechnological innovation, where we have plenty of omics data, we have plenty of clinical data, and they are waiting to be approached in a data science oriented or a data science related approaches. And in this program, we will ensure that you get a taste of how this is done by taking data from different sources, and then we will perform analysis on the server associated high performance computing server, the server Harvard Bioinfo server, and then we will show you how to make the associations from the omics data to the disease biology based on our analysis on this data. So we will also learn about basic and translational research in, for example, oncology, where the patient-derived cell lines or xenograft models and clinical trials offer a rich collection of data to for example, help us treat cancer in patients and help us give informative diagnosis at the patient level. So this, as I mentioned, has completely changed, and it has given us a complementary information approach towards disease biology, where we have traditional biological hypothesis-based approach can help us validate once we have identified specific markers and specific therapeutic responsive agents, elements that we can use in the traditional approach to verify, and traditional approach to build extremely targeted assay-based methods to understand different, for example, understand personalized and precision medicines reach to cancer patients who are responding differently to different treatments. So how do we go about this? For example, as I already introduced you, so we have to analyze, and we have to, for example, consider data from different sources, like from different molecular sources, from different even different sample sources, like microbial composition of our bodies and the environment that it is actually thriving in, and how they are responsible for destabilization of the immune system, and that leads to clinical manifestation of disease specific to the microbial abundance in specific parts of our body. And we can also relate or integrate cellular omics data with clinical phenotypical data, which is done at several different integrative levels that can be associated with common diseases, and that can also be associated with specific and rare genetic diseases, and that can be associated with cancer also in many different situations. Right? So, do we have promise to develop new methodologies and new informative approaches to data that is already been present, or do we have to resort on generating new data? But we can see from this graph that there is an information explosion, and there is an increased reliance on the data due to the lowering of costs that generate these detailed and large data. So, however, there is an ever increasing or growing information lag that complicates and the translational approaches of this data into practical and impactful insights at the point of care. So this is where, or this is what we call the analysis gap, and this is proportional, or this depends on our ability to generate new data and our ability to process, digest, and analyze the existing data to interpret it for the benefit of the individual patients. So existing data, such as data repositories from the Cancer Genome Atlas or data from NCBI, can be used to find novel approaches, and can be used to find novel biomarkers, or can be used to find biomarkers specific to specific ethnicity and race and demographic classifications of data. So what we actually address in this program is to, wait a second, So what we want to address in this program is to close the gap by focusing on the central issues that define the precision medicine, right? We will offer you training, support, and effective tools to manage, process, and analyze, and also to integrate the data so that we can make a lasting impact, and we can find novel associations, and we can at some day change the future of medicine. So our first headache, or our first challenge, is what do we do to analyze this data? In order to analyze this huge volume of data, one has to resort to the methods, or one has to address the computational complexity of analyzing this humongous volume of data. For example, a genomic data of a single cell can reach the sizes of tens and tens of GB, if not hundreds of GB, right? So what do we use to do, or what do we use to perform analysis on these data? Right? And this is just one of the major challenges that is related to, for example, omics data analysis, to accurately and to effectively process large-scale data sets that represents gigabytes, sometimes even terabytes of data. So such large quantities of data can actually prevent many of us to gain practical and hands-on understanding and hands-on feel for what this data represents and what are the patterns of this data, right? So let us quickly have a demonstration, or quickly have a hands-on overview of various methods of analysis for example, to help you overcome these major limitations. So this is how we will do this also. Let's go back. Yeah. this is one of the addition, or this is one of the pipelines that you will set up in order to understand, or in order to associate transcriptomics data with different tumor subtypes, specifically considering breast cancer in this case, right? You will get access to hands-on exercises. You will get access to the data sets that we used for practice, and these data sets are curated so that the analysis will run quickly. And you will also get access to where we got this data, to the publication that from where we got this data. And these data and several steps that involves in processing this data. For example, we have to pre-process this data. We have to perform the analysis by mapping this genomics data or transcriptomics data to the reference genome, human reference genome. And in the end, we have to identify variants in our data, or we have to identify, or we have to quantify the expression levels of different genes by including appropriate analysis methods for setting up the pipeline, let's say for transcriptomics data analysis or for genomics data analysis. So one of the results that you will get is such as this, where we can identify genes that are differentially expressed in the diseased state compared to the normal state or normal tissue. This can also help us identify genes and pathways that are enriched, for example, in a tumor tissue compared to the normal tissue that is obtained from the same patient, or in a tumor tissue and data from normal tissue that is obtained from healthy individuals. So we will learn about some of the important projects and some of the data that it collects, and the Cancer Genome Atlas is one of the very important repository that has a collection of large quantities of several different omics data for over 30 tumor types. We will get a detailed introduction to navigating this repository, finding important analysis, finding important data from this repository. We will download relevant data from this repository and perform analysis pertaining to precision medicine to understand how this repository can be leveraged to perform a project in precision medicine, let's say. On the other example, or on the other hand, we will also deal with data from patient-derived cell lines. So these are data that are generated from under one roof or from a laboratory, where they consider several numbers of patient-derived cell lines and characterize these cell lines for multiple omics data types. So the advantage of using a model system like a cell lines data over patients' data that is observed, or that can be obtained from Cancer Genomic Atlas, is that these patient-derived cell lines can be subjected to several analysis, several experimentation methods. For example, in this study, these cell lines that are derived from the breast cancer patients are actually first characterized at different molecular level, and also studied for their responses to 90 different drugs, right? So now we have data that characterizes these cell lines or tumor at multiple molecular omics level, and also we have data of their response to several different treatment options. So by integrating these two data, we will get how we can effectively identify specific molecular characteristics that can be associated with a positive or a sensitive response to a therapeutic option, or negative or resistive response to several therapeutic options. Then we can use these biomarkers and apply that to patient data from TCGA, for example, to identify or to stratify patients who are most likely to respond to the treatment options that we have identified from this study. So this example project will also be dealt with in tremendous detail in the upcoming program. So this is, for example, if we have to identify and associate the omics data to the clinical data, this can be done using data that we download from the Cancer Genome Atlas, as we have been going through. In this case, we have an example project that deals with liver hepatocellular carcinoma, and how various clinical data available on those patients, for example, their risk factor and their demographic information, and their, for example, disease history in terms of hepatitis virus and their complications, can be associated with the molecular characteristics or molecular omics data, like mRNA expression or gene expression data and microRNA expression data. So these association and integration can be made in this project, and this will also be discussed in detail in this program. So we will, in addition to this, look at, for example, phylogenetic tree reconstruction to understand the spread of Ebola virus outbreak in, for example, around 2016. And because we don't want to focus only on the cancer, we want to explore the precision medicine approach to infectious disease research, where this specific branch of precision medicine is fastly improving and are being applied to several different disease states. So another example that we will consider is to study SARS-CoV-2 genome and the cell's responses, as well as we will see how metagenomics or microbiome data is associated with the disease and drug response. So these are also we will consider in this program. So for different parts, we will, this been already covered by Sri Gauri, but we will leverage the OmicsLogic resources. Go back again, it's running or it's not running. Yeah. So we will leverage these resources. For example, we will use asynchronous coursework. We will use the cloud-based analytical server where you can analyze large genomics data. And we will use, for example, code-based lessons where you can also practice coding in R and Python. And we will also use several of the project examples that uses published data or that uses data from publicly available repositories. So to proceed further, these methods, let me actually give you an introduction to some of the pipelines or some of the outputs that you can take from the server to successfully analyze omics data into multiple different analysis pipelines that is going to give you information on the phylogeny, that is going to give you information on the gene expression pattern. It will also give you information on the structural and functional aspects of the variants that you identify from variant calling methods. It will also give you, for example, help you perform surveillance-based approaches and surveillance-based analysis on publicly available resources for example, for SARS genome, for a SARS-CoV-2 study. So without further ado, let's go to server and let me show you how you can perform this analysis by setting up a very simple demo pipeline for transcriptomics data. So let me reshare my sharing. Okay, just let me know if you can see my screen once I set up the sharing. It's still working. Yes, can you see my screen? Can you see the server page?
Yes, sir, we are able to see.
Okay, so if you go to the server Bioinfo, this is a page that you will take a look at. So once you have some credentials set up, so this demo pipeline can be accessed by everybody who has a credential in server Bioinfo, server in the big data server. So you don't have to be a paid user to analyze, to perform this demo pipeline, which will give you an example or experience of setting up this pipeline. The data that we are going to consider is a cell line data that I have been talking to you about. So that analysis will come under the transcriptomics data analysis. And you can, for example, to set up the pipeline, you can drop down this box and then choose the option demo PDX project, or sorry, let's go here, so not the PDX project, demo cell line project. So if you click this, this is going to open up into the demo pipeline. And yeah, so we will start by starting. So this has the input files have already been uploaded, so you just have to follow the lead of the highlighted sections that this demo pipeline is offering you, and the choice of this analysis method you will get to know as you go through the program. So first you have to do the pre-processing. Once you select these analysis options, you will get suggestions, right? I'm getting now suggestions based on what could be my future step. So I want to perform mapping onto the genome using HiSat2, and then identify some novel isoforms based on my sample, and their expression should be quantified. And when we do this, I will want to quantify the expression levels of the isoforms and genes I have mapped onto the genome. And then I want to perform a downstream analysis or differential gene expression analysis, which will help me identify, or which will help me shortlist genes that are differentially expressed in one type of cellular or molecular tissue type compared to the other. So I will end the pipeline. And then by ending the pipeline, you will get hands-on approach, or hands-on to several of the output files that you can download. For example, we will have expression levels of isoforms and expression levels of genes that are at different normalization that are performed for different normalization. And as I also mentioned, you will get statistics of how well your FASTQ files or the reads mapped onto the human reference genome. So this will become the quality control of your mapping, and we will also get results of our differential gene expression analysis that we have performed by comparing cell lines or by comparing tissue types that are normal-like compared to the clotting load. To perform this analysis, you need to have prior biological information about the sample. So this is how you can set up this analysis, and you can access these output files. And if you download this zip file, you can have access to all the plots that differential gene expression analysis DESeq2 produces, so that you can make sense of all the data and make sense of all the outputs that you have obtained by performing these omics data analysis and downstream analysis in the server. So I urge all of you to explore this part and explore the demo pipeline